going to focus on cellular respiration. This is how we make energy, or how we get energy to combine ADP plus PI to make ATP. That's what we need. ATP from ADP and PI that were produced from cellular work. So how does this occur? Well, in a nutshell, it's really the relocation of electrons from food, multiple food sources, we'll mostly talk about glucose in this lecture, to oxygen. So there are two components to a general redox reaction, reduction, oxidation, so it's called redox. We have oxidation, which is the removal of electrons from one substance, and reduction, which is the addition of electrons to a substance. So here's our general reaction down here. We have reactants on this side, products on this side, and in this reaction, we have that X, this molecule X, is going to be oxidized because it has an electron that can be removed. So oxidation is removal of an electron, and reduction is addition of electron. So what's going to happen is this electron is going to be removed from X, which is going to be oxidized, and it will be added to Y, so Y is reduced. So here are products. X has lost its electron, and Y has gained an electron. So I always had problems remembering that oxidation is removal of electrons. And a couple of years ago, when my son was finishing his engineering degree, I was talking to him about this and saying, oh yeah, I got up in front of the class and then I get confused. And he said, oh, here's a really good mnemonic to use. Oil rig. Oxidation is losing electrons, reduction is gaining electrons. And that has helped me remember it, standing up in front of you, and hopefully it will help you during your exam or any other time. Okay. The redox equation for cellular respiration is a little bit more complex, like the same format, and it's shown here. And you should recognize this from the LBL that you just turned in. What is this molecule? Glucose plus oxygen gives you carbon dioxide and water. So these are our reactants, these are our products. So in this equation, we have glucose that's oxidized to form CO2. So glucose is going to be oxidized to form CO2. That means we're going to rip off the hydrogens and their electrons. They're going to be taken off glucose, and eventually they are going to end up on oxygen. Okay, oxygen is reduced to form water because the electrons that were taken off glucose are going to be added to that oxygen, and we're going to form H2O. So this, we're taking all these hydrogens and their electrons away, and we end up with carbon and oxygen left, so carbon dioxide. And this one, we're going to add the hydrogens with their electrons back, and that's going to be water. This happens in a long series of steps. This is just the final product. So energy is released, and ATP is generated. Is this an exergonic or an endergonic reaction? It's an exergonic reaction. That's right, and we have formation of ATP, and this is, these reactants are at higher energy than our products, so it's an exergonic reaction that we can use to form ATP. Okay, cellular respiration can be thought of as a controlled explosion. If you take hydrogen gas and combine it with oxygen gas, you will form water because the hydrogen, the electrons in the hydrogen will be drawn very close to oxygen because what property does oxygen have? It's very electronegative, so we'll bring those hydrogen, the electrons with hydrogens very close, and in fact this is a rapid explosive event that will release heat and light energy in the formation of water. Okay. Cellular respiration is exactly the same reaction, it's just a controlled reaction, where we start out with hydrogen and their electrons, combine with oxygen, and they form water. We take the electrons off the food, 2 at a time, 2 hydrogen ions and 2 electrons, and then they are fed into the electron transport chain that allows the electrons to fall down their energy gradient, basically, and at each step you can harvest some of that energy to make ATP. And so this is a controlled explosion in that we get a low energy product from combining two higher energy products. Two higher energy reactants. Okay, where does cellular respiration take place? This should be familiar from your LDL as well. Okay, look at if you haven't. Okay, very good. The majority of you got the answer right, which is C. This is to remind you that cellular respiration takes place in the cytoplasm and in the mitochondrion, not only of animal cells, but plant cells. So plant cells use cellular respiration to make ATP as well. They also do photosynthesis, but they have mitochondria by which they make ATP. Okay, so that's really important to remember. So we have three processes that you got introduced to in the LDL. The first was glycolysis, and where does that occur in the cell? In the cytoplasm, that's right, that occurs in the cytoplasm. The second step is the citric acid cycle, and where does that occur in the cell? That's exactly right, in the matrix of the mitochondrion. So here is the mitochondrion. It is an organelle and has a smooth outer membrane and a second inner membrane. So this has an organelle with a double membrane as well. We have the nucleus, remember, had a double membrane. So does the mitochondrion. So the citric acid cycle takes place in the matrix, inside the matrix of the mitochondrion. And then where does oxidative phosphorylation take place? In the inner membrane. And that's actually that the proteins and lipids that are involved in this process are located actually embedded in this membrane. And so we'll look at that in more detail later on. Okay. I also need to introduce you to the idea of how electrons are shuttled around in cellular respiration. So NADH is a shuttle molecule that shuttles electrons from food to the electron transport chain. So there's two steps here. The first is we have a dehydrogenase enzyme that strips two hydrogen atoms. Each hydrogen atom has a proton and electron. So if we take off two, we get two electrons and two protons, and they're coming from fuel. Most often we're talking about glucose, but this could come from other fuels, both lipids and, well, actually, the dehydrogenase works on this one. So, okay, so this is, we're talking about glucose right now. So it's stripping the hydrogen atoms from fuel, and it's actually taking them, and it's going to put them on a shuttle molecule. The shuttle molecule starts out as NAD+, and when you add one hydrogen atom and one extra electron to the NAD+, this forms NADH. One hydrogen ion, the H+, the proton minus the electron, is released into the solution. So let me show you how that happens. So this molecule here is NAD+. There's a bunch that I've put a black box in, you don't have to know anything about. But the top end of this molecule is where you need to understand how this works. So NAD+ has a nitrogen right here in this position, and this ring here, and it has a plus on it. And there is also a double bond between the two carbons in this ring here. When dehydrogenase strips off these hydrogens, the electrons and the protons, what happens is one of those hydrogen atoms actually gets added to this location right here. This double bond is broken and it gets added here. So this is the result. You get a hydrogen that's added, its electron and its proton. And an extra electron gets added to this site right here. So this is a nitrogen that has a positive charge. So you can add an electron. So over here, NADH, that nitrogen is no longer positively charged because an electron's been added, neutralizing that charge. Okay? So now we have this molecule that's carrying 2 extra electrons, one that's with this hydrogen and one that's tacked onto this nitrogen. And the free hydrogen ion then was released into the cytoplasm. Okay, the NADH now can move electrons to the electron transport chain. So it's taken electrons from this fuel and it's going to use this to power the electron transport chain. Okay, so NADH is the shuttle molecule. NAD plus is what the 2 extra electrons and the hydrogen atom and the extra electron are added to. And then we have a free hydrogen ion that is deposited into the cytoplasm. Okay, so in this equation describing the shuttle reaction, which reactant molecule will be oxidized? Remember, oxidation is losing electrons. Okay, you guys are quiet again today. Okay, click in. Okay, very good. The majority of you are correct. This is the right answer because the electrons that are associated with the hydrogen atoms that are pulled off, right? The proton and the electron come along. Okay. So electrons and fuels, let me start again. Okay, there are different fuels that can be used in cellular respiration. It's not just glucose. So what we've been talking about so far is a carbohydrate, where we have a carbohydrate that is then broken down by digestion into simple sugars, and those sugars can be fed into glycolysis, and that will then be converted to pyruvate that is converted to acetyl-CoA, fed into the citric acid cycle and oxidative phosphorylation that we'll talk about in just a minute. But we can also have proteins that are feeding into cellular respiration, just at slightly different places. When proteins are broken down, oftentimes the amino acids are used to produce new proteins, but they can also be fed into the citric acid cycle here, into this part of the glycolysis, and they can be converted into acetyl-CoA to produce ATP. Okay, and we can also do this with fats. Fats, remember, were a 3-carbon alcohol that was attached to 3 fatty acid chains. Those can be broken down into glycerol and fatty acids, and glycerol is fed into the glycolysis cycle here, and fatty acids can be converted to acetyl-CoA. Okay, so the energy for driving cellular respiration doesn't just come from glucose, it can come from proteins and fat. So that's important to remember. But the example we're using most often is glucose. Okay, the important thing is that it's relocation of electrons from food to oxygen, and the released energy is used to synthesize ATP. Okay, and these are the steps that occur in the mitochondria, and these are the steps that occur in the cytoplasm. Now let's do a little overview of this process. Here is the extracellular space around the cell, and we have glucose that's outside the cell. Glucose has a lot of OH groups on it, right? Just looking at the formula, you know that. So is it a hydrophilic or hydrophobic molecule? It's hydrophilic. So it needs help going across the cell membrane. So there's actually a transporter that transports glucose into the cell. And then once glucose is in the cell, it undergoes the process of glycolysis, and through that process, it's going to produce a little ATP. We'll look at that. It also is going to produce some NADH, so it's going to take some NAD+, add some electrons to it, so that can then go over and power oxidative phosphorylation. But the majority of it is going to be converted into pyruvate, and that is going to be converted into acetyl-CoA, that is then going to enter the citric acid cycle. There we're going to generate many more NADHs and another type of electron carrier, FADH. Both of those are carrying electrons to the electron transport chain that is going to be important in oxidative phosphorylation. Okay, so we're going to start with glycolysis, which happens in the cytosol, as you've already correctly identified. The level of detail that I go into about cellular respiration in the next lecture, photosynthesis, in the lecture is the level of detail I expect you to know. The book will have additional detail that I think is helpful if you read it, but don't spend time memorizing extra details. What I want you to understand is the general process. Okay, in glycolysis, we convert glucose to pyruvate. So this is a 6-carbon sugar into 2 3-carbon molecules. This generates NADH, and the NADH is going to shuttle electrons to the oxidative phosphorylation part of the pathway. And we're also going to generate free hydrogen ions that are going to be released into the cytoplasm. In this process, we have a net gain of 2 ATPs, and these are made by substrate level phosphorylation, and I'll show you what that means on the next slide. And this process requires an initial energy investment. So even though this whole process of cellular respiration is an exergonic reaction, you do, at a number of steps, have to invest energy, a little bit of energy, to get a lot of energy out. Okay, so this is illustrated here, the energy investment phase to actually convert glucose into pyruvate. One needs to first invest 2 molecules of ATP, and after you've done that, you can generate 4 molecules of ATP, 2 of NADH, so these are going to go to oxidative phosphorylation, and 2 pyruvates, and these are going to go get converted into acetyl-CoA and then go into the citric acid cycle. So all of these, we've already started now pulling off the electrons and using them to generate carriers and making a little bit of ATP. So the net from glycolysis for glucose is we get 2 pyruvates, we get 2 ATPs, we invested 2 and we made 4, so our net is 2, and we get 2 NADHs as well. Okay, substrate level phosphorylation is where we have an enzyme shown here in blue, so that's a protein molecule, right? And in the presence of a high energy substrate, which is shown here, that has a phosphate group, this phosphate group can be added to an ADP directly and form ATP. So you get products that are at lower energy than the reactants. So this is an exergonic reaction. So you have to have a very high energy intermediate here. Okay, so you can generate a little bit of ATP by the substrate level phosphorylation. And the process of investing energy and the kinds of reactions that occur are illustrated in this short animation, and you don't have to know the details, but you have to understand why investment of energy is important. Because you have to rearrange
which begins the
molecules.
Glucose, is a series of chemical reactions that can be divided into 2 main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 molecules containing 3 carbons each. The 3 carbon molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups to ADP, finally making some ATP. A couple more reactions rearrange the atoms in the 3 carbon molecules. more ATP is generated in the final reaction that yields pyruvic acid. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed along with 2 NADH molecules.
Okay. Once you get pyruvate, or pyruvic acid, a transport protein has to move this into the mitochondrial matrix. And so here's pyruvate. Why would it be called pyruvic acid? What is this group called? A carboxyl group, so it's an acidic group, right? And so this is actually a very hydrophilic molecule, and it needs a transporter to get across the double membrane, both the outer membrane and the inner membrane, into the matrix of the mitochondria. And as it does so, there are a complex of enzymes that are involved in converting this to acetyl-CoA. So the first thing that happens is this gets removed, the carboxyl group gets removed, and we have release of CO2. We take off a hydrogen and its electron, 2 hydrogens, they're electrons, and then we reduce 1 hydrogen. So we have NADH made. That is going to take electrons to oxidative phosphorylation, to the electron transport chain. And then in the presence of coenzyme A, which is a sulfur-containing compound that is found in vitamin B, then you end up with acetyl-CoA, which is shown here. So here are the 2 carbons, right? That's the same molecule, and you have this sulfur-containing group on top. And this is what is fed into the citric acid cycle. So for each pyruvate, we get carbon dioxide, we get NADH, shuttle molecule that's carrying electrons to transport that, power the electron transport chain, and acetyl-CoA. Now, acetyl-CoA goes into the citric acid cycle, and that occurs in the mitochondrial matrix. So up here on this diagram, what this is just the process we just talked about. So it's a conversion of pyruvate into acetyl-CoA. Now acetyl-CoA goes into the citric acid cycle, and what I want you to know about this process is, first, we're producing CO2. Second, we're producing lots of electron carriers. NADH, here we have 3 NADHs. Those are going to take electrons to the electron transport chain. We do a little substrate phosphorylation, so we get a little bit of ATP out of this process as well. And there's a second electron carrier that's involved in this process here, and it's FAD that turns into FADH2. Again, it accepts electrons in the form of hydrogens with their electrons, and then transports it to the electron transport chain. So it works very much the same way that NAD conversion is to NADH. Okay, so in we have acetyl-CoA, out we have carbon dioxide, NADH, FADH2, both electron carriers, and we get a little bit of ATP. So in our glycolysis, we got a little of ATP and some NADH. Here, we get a little ATP and 2 electron carriers. And now we go to oxidative phosphorylation, where we're actually going to produce ATP, but not until the end of oxidative phosphorylation. So this occurs in the inner mitochondrial membrane. In this diagram, this part is the mitochondrial matrix. This is the inner membrane space, and this would be the outside smooth membrane. So in this inner mitochondrial membrane, so this is the highly convoluted one, you have the complex, which is called the electron transport chain. So it consists of proteins, that are shown by these big blue blobs, and one down here, and lipids, one here, right there and right there. And what happens is we have NADH that comes in from both glycolysis and from the citric acid cycle, FADH2 that comes in from the citric acid cycle, and they donate their electrons. NADH donates to this first protein, and this protein takes this electron, transfers it to a slightly lower energy state, and the energy that's harvested is used to change the conformation of this protein, and that pumps hydrogen ions from the matrix up into the intramembrane space. The electron is then passed on to this lipid molecule that then transports it to the second protein. Again, a pump that pumps hydrogen ions from here to here. Passed on to cytochrome C, another lipid that then is passed on to a third protein here, and this again pumps hydrogen ions. So we've got 3 hydrogen ion pumps pumping hydrogen ions up into this region. When the electron gets here, its final electron acceptor is what? Oxygen. So oxygen, remember we start out with hydrogen and oxygen and we end up with water. Now our electron is passed on to oxygen and then it will combine with free hydrogen ions to form water. Okay? So we formed water, we have our final electron acceptor. FADH actually contributes here. Its electron goes into this protein complex, then goes into the lipid that then is moved down the chain. Same process though, the electrons are going down to lower energy states at each site, and the energy that is being harvested is used to pump hydrogen ions. So these are powering the proton pumps, and this generates a proton gradient, and we form water. But we still haven't formed ATP. That comes in the last step of this process, which is called chemiosmosis, where the hydrogen ion gradient then is been generated as a big gradient up here in the intermembrane space, and the hydrogen ions flow down their concentration gradient. They're not actively pumped, but they actually flow down their concentration gradient through a protein complex called ATP synthase, and that combines ADP and PI to form ATP. So the hydrogen ions flow through the ATP synthase and power ATP formation. Okay, so which of these provides the energy used in oxidative phosphorylation to generate ATP? And if my TAs could come down now, get set up for the demonstration, that would be good. Okay, so click in if you haven't. Okay. So, some majority, but not complete. Okay, so let's what we're going to do now is do a demonstration to help you visualize this process, and our TAs are going to help with this. And what we're going to do is we're going to follow the flow of electrons from glycolysis, forming NADH, going all the way over to oxidative phosphorylation and chemiosmosis, and forming ATP. Okay, and to do this, I need 1 student volunteer because we only have 5 TAs, and so we need 1 student to come with us. And somebody who hasn't done it before? Yep, okay. So, volunteer right here. Okay, very good. And your name is?
Anthony.
Anthony is going to be glucose for us today. Very good. And to do that, why don't you come right over here and stand and face your classmates. And Anthony is going to get, put his arms out like this, and he's going to get these 2, which are meant to represent hydrogen atoms. So what do they have? A proton and an electron. Okay, very good. So your glucose molecule, you have more than 2 of these, right? You would have a bunch of these hydrogens. But what we have in this process is we have dehydrogenase. So Lindsay is our dehydrogenase, and she is going to go over and she is going to actually rip off what to transport to the carrier. Okay? Okay, very good. And Lindsay has these, and now she's going to donate these to NAD+. And so what's she going to donate? Electrons? Okay, you can donate an electron. I'm going to donate 1 electron. And that goes on the nitrogen, right? What else is she going to donate? Whole hydrogen, exactly. The proton and the electron. And now what happens to that? Okay, so it goes into what? Cytoplasm, and did it make it more acidic or more basic? More acidic. Okay, very good. Now we have NAD plus. Is she NAD plus? So young as NAD plus she was. What is she now? NADH. Very good. Okay, and now NADH goes over to the mitochondrion where we have our electron, our members of the electron transport chain, Becca and Carrie. And Becca is the first member, and so what is Becca going to get from NADH? Electrons, very good. And what's going to happen when she gets an electron? She's going to pump what? Hydrogen ions that are in the matrix into the inner membrane space. Okay, and what's she going to do with her electron? Pass it to Carrie, lower energy state, very good. Okay, and so now, what happens? There's another electron, right? Okay, good. Now, have you pumped? Carrie, does he pump? Yes. Okay, good. So now he's increasing the concentration of hydrogen ions up there, okay? And so now, where does his electron go? To where? We have a nice round thing here. What do you think this is? Oxygen. Okay, so it goes to oxygen. Okay, and now, and now what happens to that? Now we have another electron. So does she pump again?
Yes.
Yes, okay. Passes it. Good, and he pumps again. Yep. Okay. Okay, is it water yet? No, what do we need? Hydrogen. Very good. Okay, great. Okay, now we have water. Okay, very good. Okay, but now we still don't have ATP. We have Anne is ATP synthase in our membrane. And she is going to then conduct hydrogen ions where? Where are they going to go? Down the concentration gradient. Very good. Okay, and that's going to allow generation of ATP. So she has ADP, PI, yay! Okay, so she generated her ATP, a high energy molecule. Okay. All right, so now what we're going to do is reset everything. So we're going to reset everything, starting here, here. Okay. And so now we're going to have cellular respiration occur without any narration. Go for it. Okay. That's great, thank you. Okay, now, when watching a demonstration like this or thinking about what you might want to do, we have one of these, one that we did a previous year on YouTube, so you can look at them. Draw the elements and label using arrows to show the path of electrons. That's just a suggestion. So here is my drawing that I made when I was thinking about how to set this demonstration up because the first time we did this, this is the third year we've done it, and it was because these concepts were really hard for people to understand just from looking at the book, and so that demonstration helped a lot of people, I think. And so every year I relook at this demonstration when I get ready to set it up again because it's important to review all the steps. Okay, so now answer this question again. Okay, good. Let's see. Again, if you haven't. And you actually did pretty well on this one in the first one, but a few more people now know it. 72% of the people are correct. Electrons is the correct answer. So now, those electrons in the example we just did came from glucose, right? But glucose didn't actually go into oxidative phosphorylation. And could we have gotten electrons from other places? Right? Lipids and proteins, right? And so the electrons is the correct answer because electrons could come from any of the fuel sources, but electrons are what go into power the electron transport chain. So in the next 5 minutes, what I want you to do with your group is on a card, write one sentence for each A through D that explains its role in cellular respiration. And I saw a lot of people in the last class looking through their notes. Try doing this with your group without looking at your notes first, and then look at your notes. You can look at it. I just want you to try and do it from memory. So you'll have 5 minutes to do this. Looks like a lot of people have these down. So now I want you to pass these to the aisles and your TAs will collect them. And we have a couple minutes left to do another slide or two. Okay, we have cellular respiration generates 32 ATPs per glucose if everything is basically as efficient as possible. And so remember, in glycolysis, we made 2 ATPs by substrate level phosphorylation, and 2 NADHs that we're going to be now fed into the electron transport chain. In the citric acid cycle, our pyruvate is converted into acetyl-CoA, and we get some additional NADHs here as well. And then in the citric acid cycle, we have FADH2 that gets produced as well as NADH. And these come into oxidative phosphorylation. So we have 2 ATPs here, 2 here, and the majority of ATPs come out in oxidative phosphorylation, which is broken up into 2 processes. Electron transport, which is basically pumping hydrogen ions up into this small space, the intermembrane space, where now we're getting a high concentration of hydrogen ions, and then when there's an ATP synthase there, those hydrogen ions can flow down their concentration gradient, and Anne did this nice spin when she was ATP synthase, and then showed that activation of the ATP, and that's actually how that molecule works, or actually it's a protein, a vascular protein complex. There's a bunch of proteins that are working together, and they have quaternary structures because there are many different polypeptide chains. And these ones undergo a conformational change that causes them to spin, and that is what actually allows the ADP NPI to be regenerated. And so thanks, Anne. I've never seen that one done before by a TA, so that was really good. Okay, so...
Diane, there's a question.
Yes, oh yeah.
Why is there a varying amount of ATP being made?
Why is there what?
A varying amount of ATP.
A varying amount. Oh, because the transfer is not always as efficient as it could be, right? So you don't always, aren't always able to harvest all the energy from all the molecules that you're starting with. Okay. So the last thing I have up here is pyruvate as a key juncture in catabolism, which catabolism is defined as breaking down complex molecules to release energy. And here we were starting sometimes with glucose, but you could start with higher energy or other molecules as well, proteins and lipids. And what you should be able to do is look at this part of the pathway we just talked about, which is our glucose comes in and through the process of glycolysis is formed into pyruvate, goes into acetyl-CoA, citric acid cycle, and oxidative phosphorylation. You should be able to write what is going into each of these and what's going out without looking in your notes. If you can do that, you'll have been able to play through in your mind a lot of what's going on here. Okay, and then what we're going to do next time, and we're going to save a couple of slides for the next one, is we're going to look at what happens when you don't have any oxygen. Because this process required, what was our final electron acceptor? Oxygen. If oxygen isn't there, we can't regenerate the NAD plus that we need. We can't regenerate and make the process go forward because it has to recycle. And so we're going to have to do fermentation. So that's what we're going to start with next lecture. So the last couple of slides are going to be at the beginning of next lecture.